When it comes to transformation, few feats are as iconic as the metamorphosis of a caterpillar into a butterfly or a moth. As we all learn in elementary school, a moth or butterfly egg hatches into a caterpillar or a larva, which, much to the chagrin of farmers and gardeners everywhere, proceeds to gorge itself on every leaf it can find. Then, once it has grown large enough, the caterpillar finds a safe sheltered place and enters the pupal stage, either wrapping itself in a silken cocoon or allowing its skin to harden into a protective chrysalis. Inside, it undergoes a miraculous transformation, emerging 1 or 2 weeks later as a fully formed adult butterfly or moth. But while this life cycle is familiar to most of us, have you ever wondered what actually goes on inside the chrysalis or cocoon? How does a caterpillar, a worm-like crawling creature with no wings and up to 16 legs, transform into a completely different creature with 6 legs, long antennae, and large prominent wings? Well, this process is even more insane and disgusting than you probably ever imagined, counting among the most complex and impressive feats in the natural world. The life cycle of butterflies and moths is an example of holometaboly, or complete metamorphosis, a process which includes 4 distinct developmental phases: egg, larva, pupa, and adult or imago. The vast majority of insects, including Hymenoptera, bees and wasps, Diptera, flies, Coloptera, beetles, and Lepidoptera, butterflies and moths, are holometabolic. This is in contrast to hemimetaboly, or incomplete metamorphosis, which includes only 3 developmental stages: egg, nymph, and imago. The nymph stage is largely similar to the imago, but typically has a thin exoskeleton and lacks certain adult structures such as functioning wings or reproductive organs. For example, the nymphs of dragonflies and damselflies spend their adolescence underwater before emerging and molting to become flying adults bent on world domination. Other hemimetabolic orders of insects include Orthoptera, grasshoppers and crickets, Mandatea, praying mantises, Blattodea, cockroaches and termites, and Hemiptera, aphids and cicadas. The main advantage of both complete and incomplete metamorphosis is that the larval and imago stages occupy different ecological niches and thus do not have to compete with one another for food and other resources. For example, while caterpillars typically eat leaves, adult butterflies drink nectar from flowers. Finally, certain primitive insects, namely Archaeonartha or bristletails and Zygentoma or silverfish, have anametabolic life cycles in which the juvenile is identical to the adult form but simply increases in size with each successive molt. Butterfly and moth eggs are very small compared to the adult insects that lay them, on the order of 1 to 3 millimeters in diameter. Further, in order to transform into adults, newly hatched caterpillars must consume over 27,000 times their original body weight in food in order to grow to over 100 times their original size. They are thus genetically programmed to mostly do one thing and one thing only: eat and eat and eat. Indeed, their very first meal is often the very egg they emerge from, which gives them the energy needed to find a more substantial source of sustenance. When a caterpillar reaches a certain critical size, the corpus cardiacum in its brain releases a substance called prothoracicotrip hormone, or PTTH, which stimulates the protothoracic gland in the thorax to release a hormone called ecdysone. This, in turn, causes the caterpillar to shed or molt its exoskeleton, allowing it to grow even larger. Each of these post-moltings or echodesis stages are known as instars. Interestingly, the instars of certain insects, especially Callifordae or blowflies, are central to the practice of forensic entomology. Blowfly eggs are usually deposited on a corpse 1 to 2 days after death, while the maggots hatch after 2 to 7 days. The duration of each instar stage is roughly predictable based on different factors including temperature, humidity, and the availability of food, while the stages themselves can be distinguished by certain anatomical details such as the number of spiracles or breathing holes on the maggot's body. Thus, the specific fly development stages present when a corpse is discovered, egg, larval instars, pupae, or adults, can be used to establish a rough timeline of decomposition and an approximate time of death. In any event, throughout a caterpillar's growth, it carries within it the necessary materials needed to eventually metamorphose into an adult butterfly or a moth. These take the form of dormant clusters of specialized cells known as imaginal discs, which are pre-programmed to develop into different parts of the final butterfly's anatomy, such as antennae, eyes, mouth parts, fore and hind wings, legs, and genitals. However, during the larval stage, the growth of these discs is suppressed by juvenile hormone produced by the corpus allatum in the caterpillar's brain. The level of suppression varies from species to species, with some caterpillars actually developing rudimentary wings and other structures during the larval stage, which remain hidden beneath their skin. However, production of juvenile hormone steadily decreases throughout the larval stage. By the time a caterpillar is molted around 4 to 5 times, juvenile hormone levels are so low that instead of triggering another regular molt, the release of ecdysone now compels the caterpillar to pupate, kickstarting the extraordinary process of metamorphosis. The role of PTTH, ecdysone, and juvenile hormone in insect development were discovered in 1934 by a British entomologist with a fantastic name of Vincent Wigglesworth. In a series of bizarre experiments straight out of a sci-fi horror B movie, Wigglesworth studied the molting, pupation, and maturation of Rhodnius prolixus, or the South American kissing bug, the main vector of a tropical illness known as Chagas disease. Kissing bugs were ideal for this research since they undergo hemimetabolic or incomplete metamorphosis, transforming from egg to nymph to imago with no pupal stage, and only molt after consuming a blood meal. Ordinarily, the insects molt around 6 days after consuming blood, but when Wigglesworth decapitated them just after feeding, they never molted. However, when he decapitated the insects 3 days after feeding, they molted as per usual. Next, Wigglesworth decapitated a fourth instar nymph and a metamorphosing nymph and joined them neck to neck such that they shared a common hemolymph circulation, a process known as parabiosis. This resulted in the fourth instar nymph metamorphosing into an imago prematurely. By contrast, joining a fifth instar nymph to a fourth instar nymph resulted in the former undergoing another regular molt rather than metamorphosing into an imago. These experiments led Wigglesworth to conclude that not only was molting triggered by a brain hormone, now known as ecdysone, released in the insect's head, but that there was a second inhibitory hormone, now known as juvenile hormone, which prevented metamorphosis from starting until the nymph had reached a certain size. Wigglesworth later tracked down the production of these hormones to the corpus allatum in the rear of the insect's brain. In 1942, American entomologist Carol Williams conducted a similar series of experiments using Hylophora cecropia, or the giant American silkworm. Using fine thread ligatures, Williams tied off the caterpillars' bodies at the junction between the head and the thorax and the abdomen, preventing the hemolymph, the insect equivalent of blood, from circulating between the head to the rest of the body. When this procedure was performed just before pupation, the larva failed to pupate and remained permanently in the juvenile stage. When performed just after the start of pupation, the head and thorax of the larva pupated and developed into their adult forms, while the abdomen did not, creating a weird half-larva, half-adult hybrid. When the insect sections were tied off later into the pupation process, all three pupated and matured as per usual. These experiments revealed that pupation and maturation are actually controlled by two centers in the head and one in the thorax, now known as the corpus cardiacum and protothoracic gland, and by three rather than two hormones. As discussed before, the corpus cardiacum releases prothoracitropic hormone, or PTTH, which stimulates the prothoracic gland to secrete ecdysone. This is why when the caterpillar's abdomen was tied off after PTTH was released from the brain, but before the ecdysone was released from the prothoracic gland, the head and thorax pupated and matured, but the abdomen did not. But if you think that's bizarre, buckle up because it gets even weirder. Human centipede style, Williams decapitated a number of silkworm caterpillars and joined them head to tail so that they shared a common hemolymph circulation. He then transplanted the brain of yet another caterpillar into the lead caterpillar in the chain. Incredibly, all the caterpillars simultaneously pupated and developed into regular, albeit headless adults. It is important to note in this that insect nervous systems are more distributed than our own, with multiple clusters of neurons called ganglia spread throughout their bodies. This means that for most insects, decapitation is not necessarily fatal. In another experiment, Williams cut off the abdomen of a pupating female moth and implanted it with the hormone-producing centers from its head and thorax. The abdomen duly matured and even started laying eggs. And in his final bid to win a spot in the Mad Science Hall of Fame, Williams took 3 pupae and cut them in half just behind the thorax. The first pupa he left untouched, covering the open ends of both halves with plastic sheets to prevent dehydration and infection. The second he connected with a plastic tube, and the third he connected with a plastic tube containing a movable metal ball to stop any tissue that might try to grow through the tube and reconnect the two halves. In the first pupa, the head and thorax matured while the abdomen did not. In the second pupa, both halves matured as hormones were able to freely flow through the tube, and in the third pupa, neither half matured. Williams concluded from this third result that all wounds in the insect's body had to be healed before metamorphosis could take place. Williams renamed the substance that prevents larvae from pupating, which Wigglesworth dubbed inhibiting hormone, to status quo hormone and later juvenile hormone. Williams and Czech entomologist Karel Slama succeeded in isolating juvenile hormone in 1964, while its chemical structure was worked out by Dr. H. Roller in 1967. Meanwhile, PTTH was isolated and identified by Hiroshi Kataoka in 1990. Now, before we move on, one fascinating consequence of Wigglesworth and Williams's discoveries was the development of pesticides like methoprene, which mimic the action of insect juvenile hormone. When sprayed on an agricultural field, such products prevent insect larvae from pupating and metamorphosing into adults. Instead, they remain in the larval stage and just keep getting bigger and bigger until they die. While this does not solve the problem of crop pests in the short term, after all, it is typically larvae, not the adult insects, which are the problem, by preventing entire generations of insects from breeding, juvenile hormone mimics can help control pest populations in the long term. And while methoprene is a broad-spectrum larvicide, effective against a wide variety of insects, juvenile hormone mimics can be tailored to affect only one pest species and leave other, more beneficial insects alone. Now, back to butterfly and moth metamorphosis. After experiencing a drop in juvenile hormone, the caterpillar finds a safe, sheltered place in which to pupate. Though the terms cocoon and chrysalis are often used interchangeably, they are in fact distinct structures. Cocoons are produced by many species of moth larvae and are spun from fine silky threads produced by a specialized organ called a spinneret. Indeed, the silk used in the production of luxury clothing comes from the cocoons of the silkworm moth, Bombyx mori, which was selectively bred from wild Bombyx mandarina moths 5,000 years ago in Southeast Asia. Raised on the leaves of white mulberry or Morus alba trees, on reaching maturity, the silkworm caterpillars spin a cocoon from a single thread that can range from 300 to 900 meters, or nearly a kilometer in length. If the caterpillar is allowed to complete its metamorphosis, the adult moth will release a proteolytic enzyme called cocoonase to dissolve a hole in the wall of the cocoon and allow it to crawl out. However, as this reduces the length and thus the value of the silk threads, silkworm cocoons are instead dunked in boiling water, killing the caterpillar and loosening the thread. The whole specialized process of raising silkworms, extracting the silk and weaving it into cloth is formally known as sericulture. A chrysalis, on the other hand, is produced by butterfly larvae and is formed from the caterpillar's own skin. The caterpillar suspends itself from a branch or other structure by spinning a small silken pad and hooking onto it using a special hook called a cremaster at the tip of its abdomen. A burst of ecdyson then causes the caterpillar to shed its skin one last time, with the new skin layer beneath becoming the hardened chrysalis. Like the mature insect's exoskeleton or cuticle, the chrysalis is rendered hard and watertight by the protein chitin, forming solid crosslinks with organic compounds called quinones. Butterfly chrysalids are what are known as obtect, meaning that they completely enclose the metamorphosing larva and allow very little movement. Though by far the most familiar type, obtect pupa are far from the only kind. For example, many insects, including beetles, form exorate pupa, which are articulated and allow the developing insect some degree of movement. This confers certain advantages, such as allowing the pupating insect to startle would-be predators. Other insects, like true flies, develop coarctate pupa, which are covered in a thin membrane called a puparium, formed from the outer cuticle of the final larval instar. Pupa are further divided into decticus, wherein the insect's mandibles are exposed, and adecticus, in which they are not. Thus, butterfly pupa are obtect and adectious, while those of diopsid or stalk-eyed flies are extrate and dectious. So if you come across a diopsid pupa in the wild, be careful, it may be metamorphosing, but it can still inflict a painful bite. It is at this point in our story that things start to get really wild and gross, for once the caterpillar is safely encased in its chrysalis or cocoon, it undergoes a gruesome process of decomposition and reassembly to rival any Cronenbergian body horror film. The caterpillar's body excretes special enzymes called caspases, which begin to dissolve its muscles and organs into a thick soup of proteins and amino acids. While older biology textbooks claim that the entire caterpillar dissolves into goo, we now know this is not the case, and that certain key anatomical structures remain intact inside the pupa. For example, the tracheal system remains unchanged, allowing the butterfly to keep absorbing oxygen from the atmosphere. Indeed, it typically grows bigger, a logical process given the higher oxygen demands of a flying butterfly compared to a crawling caterpillar. Much of the digestive tract also remains remains intact, though it shrinks and becomes more convoluted as the insect transitions from a leaf-based diet to a nectar-based one. And while much of the nervous system is dissolved and reorganized to serve newly emergent adult body parts, some of it is preserved as evidenced by a remarkable experiment conducted in 2008 by Douglas Blackiston, Eleanor Casey, and Martha Weiss of Georgetown University in Washington, D.C. The researchers classically conditioned 5th instar Manduca sextor or Carolina sphinx moth caterpillars to avoid the odor of the solvent ethyl acetate by subjecting them to mild electric shocks. They were then placed in a Y-choice apparatus comprising a set of plastic pipes arranged in a Y-junction, with one arm holding a container of ethyl acetate. As expected, the caterpillars avoided the smell and chose the other arm. The researchers then allowed the caterpillars to pupate and emerge, then tested the mature moths in the same Y-choice apparatus. Incredibly, despite having largely dissolved into goo and reassembled themselves from this primordial ooze, the moths retained the aversion to ethyl acetate they had learned while still caterpillars. To ensure that this adult aversion did not result from chemical contamination carrying over from the larval environment, the team tried exposing a control group of pupae to ethyl acetate and watching the pupae of trained caterpillars, but neither had any effect on the adult moth's behavior. This means that at least part of the caterpillar's nervous system and the associate memories contained within survives the pupation process. The preserved structures in question are likely the so-called mushroom bodies, which handle olfactory sensing through the insect's antennae. Initially, contrary to popular belief, caterpillars do have antennae, though they are very small and rudimentary compared to the sophisticated structures that will eventually exist in the adult butterfly or moth. Furthermore, when the Georgetown University team subjected caterpillars at the 3rd instar to the same aversion conditioning, they maintained this aversion through the 4th and 5th instars, but not as mature adults, indicating that post-metamorphic recall involves regions of the insect's brain that do not emerge until later in larval development. At the same time as the caterpillar's muscles and organs are being dissolved into goo, the imaginal discs, newly freed from the effects of juvenile hormone, begin dividing furiously, pulling proteins and other building blocks from the surrounding soup of dissolved tissue to assemble the adult insect's various body parts, including wings, eyes, legs, and genitals. Interestingly though, they appear to have many more. Caterpillars, like all insects, actually only have 6 true legs located in the thorax. The rest, located on the abdomen, are more primitive stump-like prolegs tipped with little hooks called crochets that help the caterpillar grip onto various surfaces. Even more fascinating, prolegs appear to have developed independently from true legs via the process of convergent evolution before the genes that code for them went dormant. Then, at some point in the evolution of butterflies and moths, these genes were reactivated, resulting in larvae with both types of legs. Imaginal discs start out with only around 10 to 50 cells, but rapidly divide over the course of just a few weeks to produce structures containing tens or even hundreds of thousands of cells. The growth and development of imaginal discs is so hardwired as to be largely unaffected by hormones or even by transplantation from one insect species to another. However, this can lead to some unfortunate and bizarre consequences such as antennae, legs, and wings growing out of the wrong places. Imaginal disc cells can also suffer from natural mutations in the so-called Hox genes, which control the relative position of various body parts, leading to deformations such as antennapedia, the growth of legs where the insect's antenna should be. Thankfully, however, such gruesome mutations are relatively rare in the wild. Remarkably, as soon as imaginal discs begin to grow, they are immediately attacked by the caterpillar's immune cells, which view the discs as foreign invaders. Thankfully, however, the immune system soon collapses and is dissolved along with the rest of the caterpillar's body, allowing the imaginal discs to grow and develop unimpeded. However, this curious immunological mismatch between the imaginal discs and the rest of the caterpillar's body has resulted in some truly radical theories regarding the evolution of butterflies and moths. In his 1992 book, Larvae and Evolution, and 2003 book, The Origins of Larvae, University of Liverpool biologist Donald Williamson posited that holometaboly, or complete metamorphosis, came about as the result of a rare and extraordinary hybridization event. He argued that caterpillars, butterflies, and moths were once separate species, but at one point mated with one another to form a genetic chimera that starts out as one type of creature but develops into another. In other words, butterflies and moths are not simply a different physical form of their respective caterpillars, but rather an entirely separate insect whose genetic code is carried inside the caterpillar's body, waiting to emerge. Thus, according to Williamson, the process of metamorphosis is less a process of transformation than of genetic parasitism, with the parasitic butterfly genome effectively killing and dissolving the host caterpillar and assembling itself from the remains. Indeed, Williamson argued that many key developments in the evolution of life on Earth, including the emergence of modern biological phyla, result resulted from such dramatic hybridizations rather than the slow accumulation of genetic traits as posited by orthodox evolutionary theory. While this theory, which Williamson dubbed hybridogenesis, proved highly controversial, being rejected by many mainstream scientists, it has been championed by other prominent figures, including American evolutionary biologist Lynn Margulis. However, pending further experimental investigation, the jury remains decidedly out on hybridogenesis. In any event, if it all goes well, by the end of the first week, the butterfly or moth's rebuilt digestive system will be nearly complete, while by the end of the second week, its eyes, wings, legs, and mouth parts will be well underway. Surprisingly, for much of history, the specific details of this process remained largely unknown, since the only way of finding out what was going on inside a cocoon or chrysalis was to physically cut it open. However, this typically killed the developing insect or severely disrupted its development, so only a crude sequence of developmental snapshots could be obtained using this method. However, in 2013, a team of entomologists from the University of Manchester and the London Museum of Natural History developed a miniaturized computer tomography, or CT scanner, and used it to peer inside the chrysalis of a Vanessa cardoi, or painted lady butterfly, and image its development in real time. While the process isn't perfect, for instance, the scanner can't detect certain structures, like nerves, and the heat and X-ray radiation it produces tends to kill pupae in the earliest stages of development, it nonetheless provided a wealth of previously hidden information on the complex process of metamorphosis, revealing, for example, that the caterpillar's tracheal system remains largely intact throughout. Throughout the pupal stage, levels of juvenile hormone in the insect's body continue to fall, just as they did in the larval stage. When they finally reach zero, this signals the metamorphosis is complete and that it is time for the adult or imago to emerge. The butterfly or moth secretes a proteolytic enzyme called cocoonase to soften the cocoon or chrysalis, and then uses a pair of sharp claws at the base of its forewings to break its way out. As it does, a gooey fluid typically spills out of the empty pupal casing, comprising a mixture of unused caterpillar tissues and metabolic waste excreted by the insect during the metamorphosis process. Once free, the newly emerged adult will typically hang onto its chrysalis, cocoon, or other vertical surface while its wings fill with hemolymph, expand, and harden. Then it flies off to feed, mate, and start the whole remarkable process all over again.